Our experiment was a phenotype analysis of dys-1 and hlh-1 gene in C. elegans. The dystrophin protein is located under the sarcolemma attached to an actin filament. It is found in skeletal and cardiac muscle. Dystrophin is a protein that is mutated in genetic disorders such as Duchenne muscular dystrophy and Becker muscular dystrophy. C. elegans possess one homolog gene to dystrophin, known as dys-1 gene. Scientists studying this gene in C. elegans found in previous research that worms that have null mutations in dys-1 gene show a phenotype of hyperactive locomotion and exaggerated bending. Our mutant in particular is not only a dys-1 mutant, but also an hlh-1 mutant. The double mutant shows a phenotype of time-dependent impairment of locomotion and egg laying. This phenotype is not seen in single mutants. The dys-1 gene is a protein coding gene on the chromosome 1 in C. elegans. The allele is CX18. Additionally, the hlh-1 gene is a gene transcription factor. Our project focused on phenotype of a dys-1 and hlh-1 double mutant. We hypothesized that the double mutants would have less bends per minute than the wild-type C. elegans. We also hypothesized that the double mutant would have abnormal actin filaments as compared to the wild-type C. elegans. To test locomotion, we did a locomotion assay. We used new multi-seated plates and put a single adult, either a wild-type or a mutant, and started a 3-minute timer. We counted the number of maximum extensions of turning near the pharynx, the front end of the C. elegans. We did the 3-minute timer 3 times for each individual worm. We repeated these steps from 10 adult wild-type and 10 adult mutants. On the left, you can see tracks from a wild-type C. elegans, and the right, a mutant. The tracks are a little bit different. With this data, we compiled and averaged the data for the wild-type and the mutants. And the wild-type had more body bends per minute average. In order to observe the musculature of the mutants, we did a fluorescent staining of the actin in the muscle cells using phalloidin. We washed the worms from the plate using a PBS buffer and put them into a microfuge tube. We gently centrifuged the worms to make a pellet. Then, we suspended the worms in formaldehyde of pH of 7 and incubated for 30 minutes. After the incubation, we gently centrifuged again to make another pellet, and we removed the formaldehyde. We repeated this step using formaldehyde of pH of 8 afterwards. Finally, we washed the worms with PBS buffer and then recentrifuged to make another pellet, removed the buffer, and repeated that step 5 times. Afterwards, we put in negative 20 degrees Celsius acetone and put the tube in the freezer, and came back a couple days later. When we came back, we once again washed the worms 5 more times. Then, we added 50 microliters of the phalloidin dye and incubated for an hour. Then, we pipetted off the excess and washed the worms 2 more times. Finally, we added a few drops of equilibrium buffer and incubated for 5 more minutes, removed this supernate and added a drop of anti-bleach. Finally, we observed the slides under the fluorescent microscope. On the top is the double mutant at 20 times magnification. Underneath is the wild-type marker. From left to right is the bright field, the muscle cells highlighted in red, the nucleus of the muscle cells highlighted in green, and a merged picture of red and green. The double mutant is on the left, and the wild-type marker is on the right at 20 magnification. The mutant is different than the wild-type and shows a twisted musculature. Our results show that the dys-1 hlh-1 mutant has less body bends per minute than the wild-type, and that the musculature was much different than the wild-type. This supported our hypothesis. Since there is a difference in musculature such as would be seen in human muscular dystrophy, there seems to be conserved function among organisms. The question remains, by what mechanism does a degeneration of muscle occur in C. elegans that are dys-1 hlh-1 mutants? Currently, the sequence of dys-1 and C. elegans is pending. By discovering the genome of the dys-1 mutant and comparing to the wild-type, in the future we will know what kind of mutation occurs. In humans with Duchenne or Becker muscle dystrophy, approximately 2/3 of the mutations in both forms are deletions of one or many exons in the dystrophin gene. Thereby, dystrophin deletions usually result in frame shift. Some progress has been made in therapeutics for muscular dystrophy, such as myoblast injections and stem cell therapy. However, the research is still in rudimentary stages.